So hello students, I hope all of you are doing great. This is Basavaraj Sir, your biology master students. Welcome. Good evening all of you students. First of all, good evening all of you. Right, now this is going to be the part 1 lecture of your genetics. Now students, let me tell you one thing before I start. If you do not know anything about genetics, if you do not know what is a loci, what is a allele, what is homozygous, heterozygous, what is the difference between a character and a trait, nothing, absolutely nothing, then this video is for all of you students. Because in this lecture right now, especially the first lecture, I will be going from all the way from the basics, all the way from the what is DNA to the advanced level students. So if you are ready, and if I am audible, and if I am visible to all of you students, quickly fire emoji in the chat right now if I'm properly audible and visible to all of you students. Quickly in the chat right now students. Now this chapter is called as Principles of Inheritance and Variation. Yes or no students? It is called as Principles of Inheritance and Variation, right? Can anyone in the chat or anyone in the class, right, tell me which biological unit this chapter belongs to? For example students, human reproduction belongs to reproduction, right? Plant, the sexual reproduction of flowering plants, again reproduction, right? Your photosynthesis in higher plants belongs to plant physiology, right? Cell cycle and cell division belongs to cell biology, right? So which part of biology do you think this particular chapter belongs to? Now students, before I go any further, this chapter, Principles of Inheritance and Variation, it belongs to a much more wide concept of biology called as genetics, students. It is called as genetics. All of you in the chat. So it belongs to genetics. Now let's quickly understand some few terminologies. What do I mean by inheritance and what do I mean by variation? Shall we, students? Okay. Now let me ask you a simple thing. Let me ask you a very simple thing right now. Have you ever gone recently to a wedding or any family function, students? Any one of you? Any family function have you gone recently? All of you, tell me. Don't be shy, tell me. Have you gone to any of the family functions very recently? Yes, Paki, yes, Stalin, Aishwarya, Rubik's, Kechi Musica, Ayyappa, music. Okay, Netra, Blessy, Monishta. Tell me students, has any one of you been to any of the family functions recently? Okay. If not recently, you might have gone in the recent past. Okay. Now students, I have heard this, you might have heard this, right? Every single relative which you meet in this party and everything, right? They will tell you one small thing, that is, hey, you look like your mother. Students, have you heard that? Students, have you heard that you look like your mother or you look like your father? Your nose looks like your mother, your eyes looks like your father, you are tall as your father. Students, have you heard this line in any of the following events? Yes, you might have heard. Yes or no, students? You might have heard throughout your life, students. You might have heard about this throughout your life, right? Now, why is it so? Why is it so, students? Why is it so? This entire phenomena that you are resembling, right? You are resembling your parents because of inheritance, students, because of inheritance. So, if I have to speak about, students, if I have to speak about the first topic, that is called as inheritance, students. The meaning of inheritance is that it is a process. Yes or no? It is a process by which characters in the form of traits are passed from parents to the offspring. Students, can anyone tell me what is the meaning of offspring here? Students, in this chapter, I'll be using this word offspring a lot. So all of you should know, students, the meaning of offspring is basically all the kids. Meaning of offspring is basically all the kids only, right? All the kids only. Kids are students, right? Right, kids. Kids are basically you, right? You are the offsprings here. That's all, students. Now, that is called as inheritance. Now, students, every single time I talk about inheritance, there's one famous line, okay, very famous line. It is called as like begets like. Students, has any one of you heard about this line? Like begets like. You might have heard about somewhere in coaching center somewhere. The meaning of like begets like is that the parent and the offspring are identical to each other. Now, what do I mean by that, students? Students, in nature, have you ever seen a giraffe giving birth to a hippopotamus? Students, have you ever seen in nature a giraffe giving birth to a hippopotamus? No, it is not possible. A giraffe will always give birth to a baby giraffe, that is small cutie giraffe. Okay, a hippopotamus will always give birth to a small hippopotamus. A dog will give birth to a puppy, a cat will give birth to a kitten. So that is called as like begets like. And the same applies to you also, right? Humans will give birth to a human infant or a human baby, which has similarities. That is called as inheritance. Now, students, all of you tell me in the chat right now, all of you in the class tell me, students, are you identical to your father or mother? Are you exact copy of your mother? Or are there any variations? Or are there any changes between you and your mother? You might have the nose. You might have that amazing nose as your mother, but your eyes looks very weird. Your eyes does not look like your mother. Right? Now, when we compare a parent to the offspring, there are certain changes. These changes are called as what, students? They are called as variations. Students, they are called as what? They are called as variations. So variations are nothing but the changes or the degree to which, students, degree to which offsprings differ from their parents. Basically, the differences between parents and the offspring is called as variations, students, variations. So any doubt now, students, any doubt till now? Students, let me tell you one thing. Do not worry if you do not know anything in this chapter. I am the God today for all of you. I will teach you the best way possible to my abilities. Okay. Now, students, I want all of you to understand one small thing right now. One small thing. Now, when I speak about these variations, how do you think these variations arise? What is the secret behind these variations? Anyone? You have learned this in the previous class with me in the previous chapter called as cell cycle and cell division. Students, in this chapter called as cell cycle and cell division, there was a process which happened in Packettine stage. Can anyone tell me in the chat right now? If you are my student, you should be able to tell. If you're my student, you should be able to tell what exactly happened in the case of Packettine, students. In Packettine of your meiosis prophase 1, Packettine, there was something called as what? There was something called as crossing over. So this entire variation is because of sexual reproduction, where during gamete formation, there is something called as crossing over. Now, because of this crossing over right here, these variations arises. These variations arise, students, because of crossing over. Yes, students, yes, there are some cases where mutation also plays a role, but that is very rare. That is very rare. In general, it is always crossing over or the entire recombination. Okay, entire recombination, students. I hope all of you remember the crossing over process in the Packettine, students. Packettine. Now, students, there is something called as, I will not go over it a lot. I'll very briefly tell you. There is something called as Sahiwal cow. This cow is a result of selective hybridization. Not much detail. They'll not ask you this much. Okay. Just remember, students, there is something called as Sahiwal cow. As a result of selective hybridization, we get this amazing fancy cow. Amazing. Okay, holy cow. Student, namaskar on all of you. Now, students. Now, we come to certain terminologies. Now, let me ask all of you one basic question, very basic question. Let me just check if you are smart right now. If you're very smart. It's from Punjab, yeah, students. Students, you want that information? It's given here. You can read that, students. I'll give you my notes. Don't worry, students. Through selective hybridization, domestication from ancestral wild cows, we have well-known Indian breed called Sahiwal from Punjab. Sahiwal cow from Punjab. Okay. Now, students, I'll ask you one basic question. Let's see if you can answer, students. If you can answer, students. Okay. Very basic question. What is DNA? What is DNA, students? What is DNA? It is a very important question to all of you right now. It is not one shot. It is not one shot. Half. Okay. What is DNA? All of you. Who can tell me? Who can tell me, students? Let's see who is smart today. I will recognize the person who is very smart. I will give them J in the class. I will give a shout out to them. I will write their name on the board also if possible. If anyone can tell me what is DNA? DNA is genetic material, true. DNA being genetic material is the function of DNA actually. It is the function of DNA. DNA being genetic material, it can store things, it can pass on things, that is a genetic material. Someone will say, students, some of you will say DNA is deoxyribonucleic acid, but that's just the full form. Yes or no? So what is DNA, students? What is DNA? Is DNA a protein? Is DNA a lipid? Is DNA carbohydrate? Is DNA enzyme? What exactly is DNA? Who can tell me, students? Who can tell me? What is DNA? This is a basic question. Exactly, Aishwarya. Amazing, Aishwarya. DNA is a nucleic acid. Very good, students. Very good. So we will go from very basic right now. DNA right now, students. DNA called as deoxyribonucleic acid, which all of you know as your genetic material. This genetic material right now is nothing but a nucleic acid. It is nothing but, students, it is nothing but a nucleic acid. Okay, it is nothing but a nucleic acid. Now our next question arises, students. Next question arises. So I was lost in my thought. This nucleic acid right here, right, is a type of micromolecule or a macromolecule, students? Macromolecule or micromolecule? If you know the chapter of biomolecules, you should be able to tell me, students. You should be able to tell me, right? Right. Students, you should be. You should be able to tell me, is it a biomacromolecule or is it biomicromolecule? So students, you will tell me it is a biomacromolecule. Now, if it is a biomacromolecule, technically it is supposed to be a polymer. Now, polymers are made up of monomers. Polymers are made up of monomers. Now, what is the monomer of DNA? Students, who can tell me? What is the monomer of DNA? Students, again, it's a small reminder for all of you students. Quickly smash your like button. Smash your like button by giving me attendance, all of you. And also some hearts in the chat because my head became a little light there suddenly. Head became light a little. Okay. So, the monomer in the case of your DNA is something called as a nucleotide. It is something called as a nucleotide, students. Nucleotide. See, students, I'm going you very basic. Okay, it is not a protein. It is not a protein, students. So DNA is genetic material, nucleic acid, biomacromolecule. Molecules basically have the polymers. Polymers are made up of monomer, and the monomer here is called as nucleotide. Nucleotide. Okay, and this nucleotide mainly has three parts. Okay, it has three main parts. One is called as your nitrogen base. Students, can anyone tell me what is nitrogen bases? There is something called as ATGC. All of you might know. All of you might know there is something called as your A, T, G, and C. Adenine, thymine, right? All of you know ATGC, right? Now, apart from ATGC, students, there is something called as your pentose sugar. Pentose sugar. In the case of DNA and RNA, there are different type of sugars. All of you know. Deoxyribose sugar and ribose sugar. I will not go very detail in that. Okay. The last component is your phosphate group. Now, students, any doubt? Students, I'm going as basic as possible to make sure all of you on the same page with me today. Okay. So, students, there is something called as your DNA, genetic material, nucleic acid, biomacromolecule. It is a polymer. Polymers are made up of monomers. Monomers here is nucleotide. Nucleotide has three parts. Done. Now, this right here is a genetic material called as DNA. DNA, students. Now let me just draw a DNA for you, students. Let me just draw a DNA for you. Okay. Now, this right here is a DNA. Okay. This right here is a, students, let's say this is a DNA right now. Okay. Now, in this DNA right now, students, if I take a small segment here, small segment of a DNA can be called as what? A small segment of DNA can be called as, students, gene. So in a particular DNA, a small segment can be called as a gene. So students, if I ask you a question right now, where is gene present? What is your answer? Genes are present on the chromosome, yes, but genes are present on DNA also. It is a small segment of DNA is called as a gene. Now students, if I take this DNA right now, okay, now I will take this DNA which is right here. Now I will start to coil it around a protein here called as histone protein. This side is histone protein. Let's say I have histone protein here. I will take this DNA and start to coil around it. Okay, I'll start to coil around it. We'll do coiling. Now students, this right here is histone protein. This is your histone protein right here. Histone protein. This right here is your DNA here. The same DNA here. Now students, these two structures are called as what? Now students, these two structures now which has DNA and histone octamer, that you learn in molecular basis more. Now this entire thing is called as nucleosome, right student? This is called as what? This is called what, student? This is called as nucleosome right now. Okay, this is called as nucleosome. Now students, now I will try to just draw this one more time here. I will draw this one more time here. Okay, let me just draw this one more time here, students. Okay, now here we have the histone everywhere, nucleosomes. Students, how does it look like? Students, can anyone tell me how does it look like, students? Students, can anyone tell me how does it look like right now? Does it look like beads on string? Yes or no, students? Doesn't it look like beads on string right now? All of you. Now this structure here, the same DNA, when it has gone under coiling around histone, this right here is called as what, students? This right here is called as chromatin. Now we have chromatin, students. Okay, beads on string, and this is chromatin right now. This right here, students, is chromatin. Okay, now students, now what if I take this chromatin? What if I take this chromatin, right, and I make it undergo coiling, and I'll start to condense it? What do I get? When I take chromatin and I start to undergo condensation, condensation of chromatin will give you what? Condensation of chromatin will give me a structure like this. In the middle, students, right in the middle, it has something called as your centromere. Right in the middle, there's something called as a centromere right now, and this entire structure right now is called as chromosome. And these two are called as what, students? They're called as sister chromatids. They're called as chromatids. This is a chromatin. This is a chromatid. Now students, do all of you know? Students, do all of you know right now how do we actually get chromosome? All of you? Now students, on this chromosome, can I tell we have DNA here? We have DNA. On this DNA, can I tell right here? On this DNA, can I tell right here? I can have something called as a gene. Yes or no, students? I can have something like something have gene here. I can have a gene here, students. So this is the basics which all of you need to know. If you have any doubt, quickly ask me right now, because this chapter, if you do not understand the basics, there's no point in going forward. Okay, students. Now, DNA, genetic material, nucleic acid, biomicromolecule, polymer, monomer, nucleotide. So DNA is made up of nucleotide here, right? This right here is your DNA, student. This right here is your DNA, deoxyribonucleic acid here. Small segment is called as a gene. If I take this DNA and I coil it under around histone octamer, we get a nucleosome, and we can find a beads on string structure here. This is called as chromatin right now. If you take chromatin and if you undergo condensation, we get something called as chromosome, students. We get something called as chromosome. Now, students, let me just show you one amazing video, amazing animation to get a better understanding, students. Now, students, is the board visible to all of you? All of you. See this. Here we have the DNA here. You can see, students, all of you can see the DNA. Nice long DNA. We have your nitrogenous bases right in the middle. Okay. Now, students, if I take the DNA here, and this right here is the histone octamer here. Now, DNA is undergoing coiling here. Now, this, students, this structure here is called as your histone. Now, when together with DNA and the histone, this structure is called as nucleosome. Now, when we have multiple nucleosomes, students, can you see? We have multiple nucleosomes, students. This is solenoid model here. Then eventually, students, what do we get? We get thread-like structure here. Can you see, students? We get a thread-like structure here that is called as chromatin. Now, chromatin, if it does condensation, let's coil the chromatin, students. Let's start coiling the chromatin. If you start coiling the chromatin, we end up getting the structure called as chromosome. Now, students, is that basics clear to all of you, students? Is that basics clear to all of you? Basics about DNA. Perfect. Now, let's go ahead, students. Let's go ahead. Okay, let's just go ahead, students. Now, a question arises: do we see, students, bacteria present in DNA, students? Bacteria has DNA. It doesn't have DNA as such, but it does have genetic material which is very scattered because it doesn't have nucleus, right? Now, students, now this particular structure called as your chromosome. Do you find chromosome all the time? No. Chromosome is found only during cell division. During the cell division, we find chromosome. Otherwise, general we find chromatin. Even during the interphase, we find chromatin. Okay. See, students, here. So, dude, mitosis started in 5 minutes ago. I can't believe you still cannot condense yet. So, this person is in interphase right now. This person is mitosis. So, once mitosis is there, whenever there is a cell division, we get condensed form. When there is no cell division, genetic material is present as such. Okay. Now, students, how many of you know the difference between a character and a trait? Raise your hands. Students, raise your hands. What is a character and what is a trait? How many of you know? Raise your hands, students. If you do not know, quickly ask me right now, students, because in my class there is no hesitation. All of you should understand what is character, what is a trait, what is heterozygous, homozygous, everything in my class right now only. Now, the first concept I'll teach you right now, what is a character? What is a character, students, right now? Character. Students, please understand, students, character is nothing but, students, it is a feature of an individual. Character is basically a feature of an individual. Now, what is a trait then? Trait is nothing but, students, this particular feature is the right feature. The feature and its variations becomes the trait. Now, what do I mean by, students? What do I mean by that? Okay. Students, can any of you tell me the different types of eye colors? All of you know, students? Eye color. Now, let's take eye color. Okay. Let's take skin color, plant height, okay, flower color, Now, students, all of these these are basically features of an individual. Yes or no? These are called as what? Characters. Now, this particular eye color, it can be a blue eyes, It can be your Sharingan, like your red eyes. It can be your brown eyes. Yes or no? Now, skin color can be very dark skin color. It can be light skin color, or it can be brown skin color. Plant height, it can be tall plant, it can be dwarf plant. Flower color, it can be red color flower, it can be white color flower, it can be pink color flower also. Yes or no, students? Now, all of these are basically the variations of this feature. Yes or no? This is a feature here. This feature has variations. Now, these variations are called as what? All of you in the chat. All of you in the chat, students, tell me in the chat. Every single one in my class, tell me. All of these different variations are called as what? They are called as traits. They are called as traits. So, students, these are called as traits, and this is called as a character. Students, if that difference is clear to all of you, I am happy. That's all. Okay, students, please smash your like button if that is clear to all of you. Okay, if that is clear to all of you, students. Okay, now, students, the next thing I want all of you to understand is what is phenotype and what is genotype. Very simple, very simple. Okay, what is a phenotype and what is a genotype? Phenotype is the external morphology, technically. External morphology, how a plant or how a human looks from the outside, the muti on the outside, now the face, that is called as your phenotype. Then what is the genotype? Genotype is the genetic makeup. Genotype is the, one second, the pen fell down. Stupid pen. Oops. So genotype is the genetic makeup. Okay. Now, students, what do you mean by, next concept is, what is homozygous and what is heterozygous? Students, how many of you seen twins in real life? Students, how many of you seen twins in real life? Raise your hands. Now, imagine if there are two twins. There are two twins. Okay. Oh, students, 1 second, students. I almost made a mistake. I didn't teach you what is the allele only. Okay. Now, students, before we go forward, please understand what is an allele. Okay. Now I want all of you to understand, not by heart, what is an allele. Okay. Students, please understand. Now, let's say, students, I have one chromosome here. Okay. Now, let's say I have one more chromosome here. I have two chromosomes, students, two chromosomes. These are two chromosomes. Okay. Now, students, these two chromosomes, they look alike. These two chromosomes are identical to each other. They're like the best friends ever. They're identical to each other. Their behavior is same. Everything is same. Now, I will call these two chromosomes as homologous chromosome. I will call them what, students? Homologous chromosome. Okay. I will call them homologous chromosome, students. Now, in this homologous chromosome, let's say I have a gene for plant height. Plant height, I have a gene here. Okay. Now, let's say, students, I have one more gene here for, let's say, flower color. Flower color. Okay. Now, students, this particular same gene will have a alternative partner in crime here. Now, this is a gene. This is a gene right here on this chromosome. This chromosome has this gene here. This gene has an alternative form on a homologous chromosome here. Same location. Students, please remember, fixed location on a chromosome is called loci. It is called as what? It is called as loci. So on this loci right here, on this loci, if I have a gene here, on the same loci, homologous chromosome, I will have an alternative partner here. Now let's say here I have one gene. This gene has an alternative form here. Alternative form here, students. Alternative form on the homologous chromosomes. Now students, now what do I call this alternative form, students? Is the voice breaking, students? Sound check in the chat, all of you. Students, is the voice breaking to all of you in the chat? Students, what do I call this alternative form? Can anyone tell me, students, what do I call this alternative form of this same gene? See, here I have a gene here, students. Here I have a gene. The same gene has an alternative form on a homologous chromosome. What do I call that? Anyone? Anyone? Students, what do I call that? What do I call that, students? What do I call that? Students, give me a minute. Students, what do I call that? If this gene has an alternative form, alternative form of a gene is called allele. So that is the allele, students. That is the allele. So alternative form of this gene is called as a allele. Students, did all of you understand or not what is allele right now? Students, do you think the allele can be this? Is it possible that this gene's allele can be present here only? Is it possible to have the allele here only? Never. It can never present. Alleles are present on a homologous chromosome, opposite side, students, opposite side. Now that is called as an allele, student. That is called as an allele. Now, students, if these two alleles, try to imagine allele as your twins. Now, if the two twins look like each other, if the two twins are identical to each other, what do you call that? Anyone in the chat can tell me. If the two twins are identical to each other, students, if the two twins, if the two twins are identical to each other, that is called as homozygous condition. Capital T, they're identical to each other. So capital T is always dominant, or small t. This is called as homozygous condition. Now, students, what do I call the twins which do not look like each other? One is a boy, one is a girl. If one is a boy, one is a girl, such type of twins are called as what? In this case, heterozygous condition, students. They are called as heterozygous condition, students. Now, I want all of you to understand, students. I'll show you one more image here, students. You'll get better understanding, students. Okay, you get better understanding here. Can you see? Here, homozygous condition. Here, two alleles are supposed to be in two different, students. One is here, one is here, one is there, one is here. Okay. Now, students, here you can see alleles here. Allele is present here. Image little wrong, students. Okay. Now, students, whenever there is a homozygous condition or heterozygous condition, one allele is always dominant. One allele will always express itself. In this case, capital T is always dominant. Capital T is always dominant here. Small t is recessive in nature, students. Small t is recessive in nature. Small t is always recessive in nature. Here we have both dominant, here both are recessive, students, both are recessive. So remember, students, this is dominant allele actually, not gene, dominant allele, and recessive allele is in the background, recessive allele. So recessive allele will not express, dominant allele will always express. This idea is heterozygous condition, where you can see, students, here capital T will always show dominant effect. Why, students? Right, it is always tall. This is also always tall because capital T is always dominant. capital T is always dominant. So this phenotype is expressed. But students, in this case, both are recessive and it is not going to express, students. Both are recessive in nature, students. Both are recessive in nature. Yes or no, students? Students, all of you understood the concept. What is dominant and what is recessive? Cash will come, students. Cash is little moody. Cash is very moody, students. Cash is very moody. Okay. Students, did all of you understand what is the dominant and what is recessive? Here also, this capital T is dominant. That is why it is expressing always. Small t is not expressing. No, students, no. No. So, students, with that, all of these following terms, right? Now, students, I want all of you to remember this dominant in biology can also be called as wild type. You might encounter many questions. Wild type is always dominant type also. Wild, not dominant, it is dominant. Wild type is always dominant. Students, dominant students, I always say this. In one classroom, students, are you first bencher or are you last bencher, students? Students, are you first bencher or last bencher? Right. I always tell this in my class. Last benchers are like your dominant. They will always shout. Students, they will always they will always keep shouting. Doesn't matter if small front benches are, they'll keep shouting. They're called as, right? They're called as your back benches are called dominant. Your front bench students are recessive. Why? Because they will never open their mouth. Only when the last bench is absent, they will open their mouth and you can hear them. Students, you can hear them that time. So first benches are very quiet always. Okay, they're very quiet in the class. I love the first benches. They're very quiet. But last bencher students, one headache. Students, I'll tell you why. Because I was also last bencher for a very long time. So I know how much I have done, how much of nuisance I have done to my teachers, even I know. This is like karma coming back to me. But now, once I've reached MSc, I became a middle bencher. Not here, not there. Okay, students. So, okay, done, students. Done. Okay, students, with that being said, now shall we go to the main chapter starting now? Because for the last 40 minutes, I gave you basics of everything which you need to know to understand and start this chapter. Students, shall we start now? All of you. Students, shall we start right now? Yes? Yes, I'm a fan of Rocking Star, obviously, students. I'm from Karnataka only. Students, shall we start the chapter now? Students, how did you find the basics, all of you? If you can tell the entire basics in one word in the chapter, students, how was the basics? Students, I studied from Christ University, my master's degree. My master's degree is in Masters of Botany. All of you bought army, right? Masters in Botany from Christ University, right? Students, now we will start the entire chapter right now. Okay. Now the first scientist, right? The first scientist is called as Gregor Mendel. Gregor Mendel is your father of your entire genetics. We always call it father of genetics. Now we will understand a few things about them. We will learn a few things about them, and we will understand how failure in life doesn't mean the end of everything. Okay, I'll tell you a very amazing story about Mendel. So if you want to know the story, smash your like button. Okay, now students, now Gregor Mendel was technically from Austria, not Australia. Gregor Mendel was from Austria. Austria is a different country only. Okay, in Austria country, right? He was born on July 22nd, right? Now, let me just 1 second. All this shouting has been making my students. He's your content creator of this chapter. Students, Gregor Mendel is the content creator of this chapter. So if you can tell me who is Gregor Mendel? Gregor Mendel is the content creator of this chapter only. All of you drink some water, students. All of you drink some water. Students, all of you drink some water. Okay. Okay. Now, students, Gregor Mendel was born. Different color. Gregor Mendel, students, was born on. Students, this month only his birthday is coming. All of you. July 22nd, 1822. This boy, students. His main work was on a special type of plant called as Pisum sativum. Students, what do you call, students? What do you call Pisum sativum in your language? Students, in Hindi it is called as matar. In Kannada it is called as batani. Right? So in Tamil, Telugu, whatever your language is, Quickly, students, what do you call Pisum sativum in your language? All of you. So Pisum sativum is basically the pea plant, students. It is basically a pea plant. So always capital letter, right? Pisum. Then we have small letter, students. Then we have small letter. So always remember, the generic name is supposed to be capital, specific epithet is supposed to be small. They are supposed to be separately underlined, students, separately underlined. So he worked on pea plant for 7 years. Students, you can hardly study for 1 year, dropper, you're crying. 7 years, students, 7 years. That is from your 1856 to your 1863. 7 long years, students, 7 long years he worked. Okay. Now, students, in his entire work, he worked with 7 pair of traits. Or you can call, students, if I say 7 pair of traits, can I tell it as 14 traits? Yes or no, students? Because a pair means 2. So do not get confused in examination if they tell 14 or 7 pair. 7 pair and 14 means same to same. Both meaning is same. He worked with 7 pair of traits or 14 traits and 7 characters also. 7 different characters. So 7 characters, 14 traits was his work. 7 characters and 14 students. In his entire work, students, he made something called as your pure lines. Pure lines. Now, what do you mean by pure, students? What do I mean by pure lines? Pure lines are your type of plants which has undergone repeated self pollination. It has undergone, students, repeat J.J. Mendel. Okay, he has worked on your 7. He has mainly found pure lines. Pure lines are your types of your plants which has gone repeated self pollination. Now, what do I mean by pure lines? Pure line means homozygous condition. It means what, students? Pure line means what? All of you in the chat, pure line means homozygous condition. All of you know, right? It can be capital T or capital T, small t, always homozygous conditions. So he worked with homozygous conditions, students. Now, students, now let me tell you one small interesting story about Mendel. Do you want to know, students? Do you want to know why you should never quit in life? Yes, students, do you want to know a very inspiring story? Mendel initially did not want to be biologist. Yes, he never wanted to be a biologist. He was a priest and his main field of interest was mathematics. It was mathematics. But unfortunately, when Mendel gave the examination, right, he failed. He failed in mathematics. To become a teacher in mathematics, he failed miserably. When he failed, he came back to his monastery, the entire church and everything, right? There, initially, as a curiosity and a hobby, he started planting the pea plants. But because of the observations he made, because of the observations he looked around himself, he realized there is a pattern. There is a continuous, continuous pattern which he observed, and that led to the genetics. Students, try to imagine what would actually happen if Mendel was not observant, if Mendel was very lazy, if Mendel gave up. This chapter would be deleted. This chapter would have been deleted from your syllabus. But apart from that, we would not make the advancements in genetics. Not possible. Okay, not possible, students. So he was the starting scientist, students. He was a starting scientist who actually used mathematics in biology. So if you want to scold someone to find numericals, if you want to scold someone to bring numericals in biology, here is the person. Here is the person. Okay. So he also applied maths. He also applied maths in biology. Crazy, no? He also used large sample size. Large sample size. More than 10,000 plants. Students. More than 10,000 plants. Okay. Now students, I will teach you exactly the characters and traits taken by Mendel. Here we go. Okay, students, we see here. Now these are all the characters. Can you see? Seed shape, seed color, flower color, pod shape, pod color, flower position, stem height. All of these are basically the characters. Now students, this character will have a alternative form. Yes or no? It will have alternative forms. So this character has two traits: round wrinkle, yellow green, violet white, full constricted, green yellow, axial terminal, stem height we have tail, tall and dwarf. So here we have characters, here we have traits. Now students, if I ask you a very simple question, students, if I ask you a very simple question, how many characters and traits did Mendel take in his experiment? What is your answer? I'll tell you why he took pea plant only. Students, do you want to know why Mendel was lucky also? Students, do you want to know why Mendel was lucky? Okay. Students, do you actually think Mendel did not work on other plants? Like, do you actually believe that Mendel worked on onion pea plant? Not at all. Not at all. Not at all, students. He worked on hawkweed. He worked on beans. Right? When he worked on other plants, do you know what happened? He did not get the same result. Mendel could not replicate the same result in other plants. So Mendel was lucky that he chose pea plant. Why was he lucky? If you want to know, stay tuned on the class till the very end. When I teach you linkage and recombination, that time remind me, I'll tell you why Mendel was lucky. Okay, why Mendel was lucky. I'll tell you that time, students. Okay, if you want to know. Why Mendel was lucky. Okay, because when Mendel did the same experiment, he did not get the same result. He never got the same result after that. Different plant. Okay, now students, now here are all the characters. Here are the contrasting traits. Now sometimes in exam, students, right, students, obviously I was test. Obviously, students, right? Okay, now right now sometimes in examination they can ask you which of the following are dominant traits and which is a recessive trait. So for that, there is a small trick called as gravity. Again, students, not my trick. Honestly, I took it from internet. I saw on the internet. I'm like, oh, this is very nice. So I took it for my class. Okay, not my trick. Okay, it is called as gravity, students. It is called as gravity. It is called as, students, gravity. Now, what do I mean by gravity here? Now, G for your green. G for green here. That is basically your pod. Pod color. Okay, that is basically your pod color. Okay, then students, then we have R here. R is basically round. Now, round, students, where is it? Seed shape. Seed shape. Okay, then students, then I have your A. A is axial. A is nothing but axial. Students, where do you see axial here? Right here. It is flower position. Okay, so then what do we have? V. V is violet here. Can you see, students, violet anywhere? Violet here. That is flower color. That is your flower color. Okay, then students, then we have your I. That is inflated. Inflated, friends. So inflated is pod shape. Okay, T is obviously tall, plant height. T is tall. That is the plant height. Now, what is Y, students? Anyone? What is Y here? Students, can you see yellow here? Yellow for here. Seed color, yellow. Yellow is your seed color. Students, with that, here we have all the students. Here we have all the, right? Here we have all the dominant students. Okay. Here we have, not student, I've stole this trick, not mine, student, not mine. This, I stole this trick. Okay. So I stole this trick. So here we have all the dominant traits. All the dominant traits are written here, students. Okay. Happy all of you? Happy? Okay. Happy all of you, students? Okay, students. Nice. Again, students, not my trick. Again, I stole it from somewhere. Do not know where. Okay. It is landed in my pocket now. Okay, now students. Now all of you also know. See, knowledge is supposed to be shared. Sharing is caring. Knowledge we share. Yes or no, students? Knowledge we share. Right? Then we care. Okay. Now, students. Now here we have, why did Mendel take the pea plant only? You can arrive. You can ask me, why didn't he take apple? Why didn't he take mango? Because, students, mango and apple are perennial. They will take a lot of time to give results. So they needed an annual plant which can keep on multiplying very fast. They need a herb generally, right? So pure varieties easily available here. That is, pure varieties are easily available. Dominant recessive, large number of seeds produced per plant, lot of seeds produced, students. Okay. No, sir, I'm not happy. Students, be happy. Why you're not happy? Be happy in life. Easily detectable contrasting characters here. You can easily tell, students. You can easily tell tall, dwarf. Everything can be told easily. Everything can be told easily, students here. Now, if you notice here, all of these are pure lines. All of these are pure lines. Remember, students, they are pure lines. All of them are pure lines, students. Okay. Now, students. Now, short lifespan, reproduces in 3 months, can be grown easily. So here are all the reasons why Mendel took the pea plant. Done, students. Now, from Mendel's experiment, we get 3 laws. Students, can anyone tell me, if you are smart enough to tell me, which law here is universal and which 2 laws always have exceptions? What tell you, students? What tell you? Apple is already used by Newton now. Okay, students. Newton has done patenting. Every day nahi leta hu main. I take alternate days. I take once class 11th, one class 12th. Science world. That's how we will do it. Okay. Now, students. Now, which of the following 3 laws here? Ta-da! Which law here is universal and which 2 laws have exceptions? They're like, these 2 laws have exceptions. They are like, we can easily discard them, students. Okay. I'll tell you, students, test cross Sanjana. Wait, Sanjana, have some patience. I'll tell you. Okay. Students, out of these 3 different laws here, right? Law of dominance, law of segregation, law of independent assortment. Only law of segregation is universal. Doesn't have any exception. But law of dominance have exceptions. That is your incomplete dominance, codominance. And law of independent assortment also has exceptions. This was shown by T.H. Morgan, which I'll teach you later on, students. Okay. Which I'll teach you later, students. So both of them have exceptions. The only universal boss here, Chris Gale. Okay. So the only universal law here is law of segregation here, students. Law of segregation. Okay, students. Now let's understand what was Mendel's work actually. See, this Mendel, that Mendel is okay. What was his work? Actually, his work was known as hybridization. And in hybridization, we have 3 main steps. I'll tell you why it is universal also. Okay. I'll tell you why it is universal also. Wait a minute. So it has 3 main steps. The first step is called as identification of parents. Okay. Or selection of parents. Second step is called as the cross-pollination. In this cross-pollination, we generally get the F1. F1 is called as first filial generation. Okay. In third step, what do? We do selfing of F1, students. Students, these notes will be available on Telegram channel. All of you students, right? These notes will be on Telegram channel. You can download from there. Okay, students, which team do you support, students? I support RCB because I'm from Bangalore, obviously. Right. Now, there are 3 steps of hybridization here: selection of parents, cross-pollination, selfing of F1. 3 main steps here. Now, students. Now, shall we start the monohybrid cross? Students, shall we start the monohybrid cross? All of you drink some water. Students, all of you quickly smash your like button. All of you quickly smash your like button, all of you. Students, let's start with the first now. No, students, no. Students, let's start with the inheritance of one gene right now. Inheritance of one gene is basically the monohybrid cross. It is basically the monohybrid cross, students. Now, students, do you want DPPs for this chapter? All of you in the chat quickly. Do you want daily practice questions from this chapter? Whatever you have learned till now in my class today, do you want DPP for that? For that, DPPs I'll give you in my Telegram channel, right? Or in this description, in description of this video, I will upload a special link. From that link, you can click and get the DPPs for free of cost. Free of cost, you will get the DPPs for this particular session today. Okay, it will be on my Telegram also. The my Telegram is Basavaraj Sir Biology. All of you know, right? All of you know, right? Basavaraj Sir Biology. Type my name, you will get my Telegram. They are also be uploading it, my DPPs. Okay, so don't worry, students, don't worry. Now, inheritance of one gene is very simple. First thing we do is write parents. First thing we do, students, is write parents here. Okay. Now, students, here who are the parents, students? The parents are basically your homozygous tall crossing with homozygous dwarf. Okay, here we will have capital T, homozygous tall, small t. Okay, now students, next step is called as gamete formation. Here we get one gamete, here we get one gamete, small t and capital T. Next step, student, next step is called as your F1. In F1 we take this and we take this and we cross it. So here, students, F1 we will get is capital T, small t. Now, students, the question arises, what is this capital T, small t look like? Will this plant be tall plant or will this plant be dwarf plant? All of you in the class tell me. Students, all of you in my class tell me, students. All of you in my class quickly. All of you. Capital T, small t is basically, capital T is dominant. So this is going to be tall plant. Tall plant. Done. Now, next step is called as selfing of F1. Okay. Here what we do is capital T, small t cross with capital T, small t. Here we do something called as Punnett square. Students, all of you know about Punnett, the scientist Punnett. All of you, have you seen Punnett, sir? Students, how many of you seen Punnett square? This is called as Punnett square. Here we do male, here we do female. So male capital T, small t are the two gametes from here. This will give you two gametes. So capital T, small t, students. What do I get here? Capital T, capital T. What do I get here? Capital T, small t. What do I get here? Capital T, small t. What do I get here? Small t, students. Now, last step is called as your phenotypic ratio as well as genotypic ratio. Okay. Now, students, the phenotypic ratio is basically what? It is how a plant looks from the outside. Here, this plant is tall, this plant is tall, this plant is tall, this plant is dwarf here. So 3 are tall, 1 is dwarf, 3:1 ratio. Genotypic ratio, students, what? Homozygous dominant only is 1. Heterozygous are 2. Homozygous recessive is only 1. So your genotypic ratio is going to be 1:2, 2:1 is going to be genotypic ratio. 3:1 ratio is going to be phenotypic ratio. Okay, done, students, done. Now this right here is called as your monohybrid cross, students. Monohybrid cross. This is your Punnett sir, right? Let's see Punnett. He was a scientist who gave us the entire checkerboard. This is also called as Punnett square or checkerboard, right? Or checkerboard. Okay, students. Now here is again a cross, students. Here is again cross. Now, students, here is a simple question. Let's see who can tell the answer, students. I hope it is visible to all of you. Students, I hope it is visible to all of you. If it is not visible, let me just read it for you, students. If it is not visible, okay, let me read for you, students. Right. Mendel studied 7 pair of contrasting traits. See, 7 pair means 14 traits. 14 traits. In pea plant and proceed the law of inheritance. Okay. 7 characters. See, 7 characters. Okay. 7 characters examined by Mendel in his experiment on pea plant where seed shape, flower color, right? Seed color, pod shape, pod color. Yes, it makes sense, students. It makes sense. Students, it is not, students, don't worry if it is not visible. I'll give you the notes. I'll give you the notes. You can see the question in the notes also once. Okay. No. Students, unfortunately, all these questions are very tiny. I don't know why. Okay. Students, let me just, I'll read this question also, students. I hope this is visible to all of you. A cross between tall plants was made. A cross between tall plants was made, resulting in an offspring of tall and a dwarf. So you're taking 2 tall plants and we crossing, and we get tall and dwarf plant in 3:1 ratio. The genotype of both parent is going to be, the genotype of parents is going to be, students. I'm going to move aside, read the question once. All of you, read the question. All of you read the question, students. Done? Students, all of you read the question. Done? Students, if you see here, it says a cross between tall parents. So here, this is the dwarf parent here. This is gone. Dwarf parent, this is gone. Okay. These 2 options are gone here completely. Okay. Now, if you look at these 2 options, let's take this option first. Okay. Now, if you take capital T, right, one parent will get capital T, and we get capital T. Students, what do we get here? What do we get here? All of you? We get all tall. This is not possible. Look at this option, students. Capital T, small t, capital T, small t. What do we get here? Tall, tall, tall. One dwarf. 3:1 ratio. Yes or no? Students, we got 3:1 ratio. Yes or no? 3:1 ratio. We got option number 3 is right. Students, option number 3 is right in that case. Yes or no? 3:1 ratio. Okay. Students, see this question. Students, you will get such questions. Please remember, you'll get such questions. Please try to increase the, these are NEET PYQs. These are not my questions. These are NEET PYQs. These are NEET PYQs. Okay. A heterozygous pea plant, capital T, small t. Heterozygous pea plant, okay, we can't use capital T. Heterozygous pea plant with violet flowers, so capital V, small v, okay, was crossed with homozygous pea plant. Homozygous pea plant with white flowers, so small v, okay. Violet is dominant over white, obviously we know that. Which of the following is expected, students? What do we expect in the, right, in the combination among 40 progenies? Let's do the cross first. Let's do this cross. What do we get? Capital V, small v, small v, small v. What do we get? Capital V, small v, small v, small v, capital V, small v, small v, small v. So students, here do I get these 2 are violet, these 2 are white. So there is 50% is your violet is to 50% is white. So if you look at the option here, it says 20 produced, students. 20 produced are violet, 20 produced are white. So option 2 is the right answer. Option 2 is the right answer, students. Yes or no? Option 2 is the right answer. Very much cheating, students. Very much cheating. Okay. Students, quick answer, I'll go to the next concept. Quick answer, I'll go to the next concept, students. Quick answer, I'll go to the next concept. Quick answer, I'll go to the next option. It is called as Punnett square. Okay. Students, how many of you know what is a back cross? Students, how many of you know what is a back cross, students? Students, Cash, I will show. Cash, students, Cash is very lazy. Cash is very lazy. He doesn't listen to me only sometimes. Students, how many of you know what is back cross? All of you. How many of you know what is a back cross? All of you in the chat, quickly, tell me. How many of you know what is a back cross? If you did not know, if you want to learn what is a back cross, quickly ask me, students. So students, what exactly happens in the case of back cross technically is that in the case of back cross, we take F1, and we cross with parents, right? In back cross, we take F1 and we cross it with the parents here. Okay. Now back cross, there are two types of back cross actually. Okay. There are two types of back cross. We take F1 and we cross it with parents right now. Okay. Now there are two types of back cross actually. In one, we take F1 and we cross it with recessive parents, and in other condition we take F1 and we cross it with dominant parents. Dominant parents. Now this right here is called as your test cross. This right here is called as outcross. Outcross is not in your syllabus. This is in a syllabus. So now I will teach you what is test cross, students. Test cross. Okay. It is called as test cross. Okay. Students, dinner I will have to eat. Students, even have to eat dinner, no? Okay. Now, students, this test cross here is nothing but it is a cross between. It is a cross between F1, right? We are taking F1, right? It's a cross between, students. It is a cross between actually dominant phenotype, right, and recessive parents. Okay. Now let me just ask you one simple question, students. I'll teach you test cross like never before. You will actually understand test cross, not by heart it right now. Please listen to me, students. Sanjana is coming back here. Okay. Sanjana, listen to this very carefully now. Now, students, all of you tell me in the chat right now. When I say a tall plant, right? When I say a tall plant, what is the genotype? All of you students. What is the genotype? Okay. Why, when I say tall plant, what is the genotype? All of you. Tell me properly, when I say tall plant, what is genotype? Students, I will do it in 3 classes. 3 classes will be over. 3 classes. When I say tall plant, what is the genotype? All of you. This tall plant is the phenotype, right? This is the phenotype. This is the phenotype right now. Now, when I say tall plant, it can have 2 genotypes. Yes or no? This tall plant can be capital T, or it can be capital T, small t. Yes or no, students? All of you. Yes or no, students? Yes or no? All of you. Now, students, we are doing test cross. We are doing test cross to find the unknown genotype of dominant phenotype. Beautiful. Yes or no, students? Beautiful. We are doing the entire test cross to find the genotype. See, this phenotype is a dominant phenotype right now. This right here is a dominant phenotype. Now, this dominant phenotype can be capital T, or it can be capital T, small t. Both are possible, students. Both are possible. Yes or no? Both are possible. To find out which is the genotype of this tall plant, we do the test cross. Now, let's assume, let's assume right now. Okay. Now, let's assume we take the dominant phenotype here. In this case, right, students, in this case, first of all, let's assume in this case right now. Okay. In test cross, what do we do, students? We take the dominant phenotype, that is basically your capital T here. It will have only one gamete, yes or no? It will give you only one gamete. Okay. So, for convenience sake, we'll write just 2 because we'll never get 2, we'll only get 1 generally. Right. And we have the recessive parents. It is a cross between dominant phenotype and recessive parents. So, recessive parents is what, students? Small t, small t. Generally, we write only 1. So, what do we get, students? What do we end up getting? Tell me, students. How are all the plants here, students? Tell me. How are all the plants here? Tell me students. Tell me students, all of you. Tell me, how are all the plants here? What is the phenotype here? So all the plants here are tall, yes or no? Okay. So if I look at this F2 generation, can I tell by looking at this F2 generation, can I tell the genotype of this particular tall plant here? Yes or no? So if I get all tall here, this has to be capital T. Okay. Look at this case, student. Look at this case right now. In this case, what do we get? In this case, we get capital T, small t, and here we have small t, small t. What do we get? Capital T, small t, capital T, small t, small t, small t, capital T, small t, and small t, small t. Students, here we get 50% are tall, 1:1 ratio. Yes or no? 1:1 ratio. So by looking at this students, by looking at this phenotype here of this F2, by looking at this phenotype here, can I tell the genotype of this dominant phenotype? Yes or no, students? By looking at this phenotype here, I can confidently say that this has to be capital T, small t. So the whole point of doing test cross is to check something. No, you write test to check your knowledge. We do test here to check which is the genotype of this dominant phenotype. Students, how many of you understood? Here? This is the NCERT diagram here. It says, again students, it is supposed to be capital V and capital V small v because violet. I do not know why they've given you W here. W is supposed to be wrong. Okay. NCERT mistake. NCERT can also make mistakes, students. NCERT can also make mistakes. Bye bye, Nandini. Right. Now here we have dominant phenotype here. Dominant phenotype, it can be capital W or it can be capital V, small w. Both are possible. Yes or no, students? Both can give you violet color here. Similarly, we had here also, no? Both can give you tall. Both can give violet. Now, what do we do, students? We cross it with recessive parents. Small w. So, by looking at this F2 generation right here, can I tell the genotype of here? Yes or no? By looking at this, I can tell what is here. Yes or no, students? Yes or no? All of you. By looking at what is the phenotype here. I have 2 violet, 2 white. It means that somewhere there should be recessive allele. That is why I am getting white here. In this case, I did not get white at all. So it says that there is only dominant allele present here and we get capital. Done, students. Yes, we do it for dominant phenotype only. Yes or no, students? Okay, students. Students, how many of you understood instead of by-hearting all this? Students, how many of you understood instead of by-hearting all this? How many of you understood this actual process of why do we do test cross? The whole point of test cross is to identify the genotype of dominant phenotype. Okay, genotype, right, of this dominant phenotype. Beautiful, students, beautiful. So imagine, students, this is so beautifully thought, test cross. Now, students, no. Now, students, I'll teach you what is law of dominance. Students, ready, all of you? Ready? Law of dominance. Law of dominance is very simple. When there are dissimilar pair of alleles, one allele is dominant, one allele is recessive. That's all, students. That's all. Okay. One allele is dominant, one allele is recessive here. That is, characters are controlled by discrete unit called as factors. See, Mendel called alleles and genes as factors. What we call genes and alleles, Mendel called them factors that time. Okay. Factors occur in pair, yes or no, students? Capital T, small t, they occur in pair. In a dissimilar pair, like this condition of factors, one member of the pair dominates. Capital T is dominant, small t is recessive. Law of dominance is used to explain the expression. It is used to explain the expression of only one of the parental character in the monohybrid cross in the F1 here. In this F1, right? In this F1 right here, we get only tall here. We do not get short. Why? Because we are getting tall here because this is dominant. That is why we are getting. Yes or no? Okay. Okay. Students, it's fine. We go slow also. We have time. But please understand. If you hurry, you will not understand. Okay. Now, students, now. Right. An expression of both F2 and explains the proportion of 3:1 ratio obtained in F2. That is law of dominance, students. Now, many students struggle to understand law of segregation. Many students struggle to understand why it is called as law of purity of gametes. Do you want to understand, students? Today, do you want to understand why it is called as law of purity of gametes? Yes or no, students? Yes or no? All of you. Many students just by heart this, but do you want to understand what is law of segregation? What is law? Why is it also called as law of purity of gametes? Do you want to understand? So if you want to understand, students, first of all of you tell me, how many of you know how to make tea? Not just drink tea. How many of you know how to make tea also? Tell me in the chat, students. How many of you know how to make tea? How many of you know how to make milkshake? Let's say we make Oreo milkshake today. Let's say we make Oreo milkshake today. Oreo milkshake. What will you add in Oreo milkshake? Tell me in the chat, students. What will you add if I have to make Oreo milkshake? In the chat. Okay. Let's say I'm making Oreo milkshake right now. If I have to make Oreo milkshake, I will add Oreos. I will add milk. I will add sugar. I will add chocolate syrup. I will add vanilla ice cream. Everything I will make. I will mix. I'll mix. Okay. Then tada! I have my Oreo milkshake. Now, students, after mixing all the ingredients, I will give you the Oreo milkshake to all of you. Now, I will ask you to tell me, students, is it possible? Is it possible to remove all the ingredients separately now? From the Oreo milkshake, can I remove sugar separately? Can I remove Oreos? Can I remove your cocoa powder, chocolate syrup? Can I remove the milk? Can I remove the ice cream from it? Is it possible, students? Is it possible? All of you? Is it possible to remove them? Tell me, students, is it possible to remove them? Not possible. Because everything has undergone blending there. The same thing other scientists during Mendel's time also thought. Whenever we get, students, whenever we get this condition, capital T, small t is present together, since two alleles are together and we are only seeing tall, they assumed, students, they assumed that this capital T was completely blending this small t and small t was lost. So everyone thought small t, capital T, both alleles are fusing. But it was our Mendel sir who told us that all of you are wrong. I am correct. I will tell you. He gave a law called as law of segregation. Law of segregation says that when two alleles are present like this, they do not blend. So the one thing, students, the first thing which Mendel uncle told us, okay, Mendel told us that, students, Mendel told us that alleles do not show blending. Okay, so if they do not show blending, what exactly happens, students? Whenever there is a two different alleles, heterozygous condition, whenever there is a heterozygous condition, they will segregate into gametes. Alleles do not show blending, they segregate into gametes. They will segregate during gamete formation. Alleles will segregate during gamete formation, students. Now, students, let's check. Let's say if alleles showed blending. Let's assume if alleles show blending, students, if alleles did show blending, how in the world do we get small t again back, students? How is it possible to get the small t back? Not possible, right? It should have been technically not possible at all to get this small t. Yes or no? If alleles were blending completely, this small t should have been lost. Then how in the world did this small t come back again? This small t came back again because alleles do not show blending. They segregate during gamete formation. They segregate during gamete formation like this. They segregate during gamete formation, students. Now, students, all of you tell me right now in the chat, this gamete will show which trait? All of you. This gamete is pure for dwarfness. This gamete here, right here, it is pure for tallness. Yes or no, students? These gametes are pure for a trait. So can I call them law of purity of gametes? All of you? Yes or no, students? That's what they are called as law of purity of gametes. Each gamete is pure for a trait. Each gamete is pure for a trait. And with that, law done. With that, law is done, students. Okay. Yes, Amrita. It explains the factors or alleles of a pair segregate from each other and gametes only one factors. Yes or no? And this particular law is universal. No exception whatsoever. No exception whatsoever, students. No exception whatsoever. It is universal. It is universal in nature. Okay. Students, that is called as the law of segregation. Students, how many of you understood? Also called as law of purity of gametes, students. Also called as law of purity of gametes. Law of purity of gametes, students. Okay. Students, I will... Students, shall we do this in 4 parts? Students, shall we do this in 4 parts? Students, shall we do this in 4 parts, 4 different parts, this chapter? Because if I go, students, if we go right now, I can finish right now incomplete dominance, I can finish codominance, and okay, we'll stop there. Students, we will stop after codominance. Shall we? Students, we will stop after codominance. Yes or no? Yes or no, students? After codominance, we will stop. After codominance, we will stop, and second part will include almost all the chapter. Okay, students. Second part will 2 is fine. 2 is fine. Actually, 3 will be too much, I feel. Okay, so 2 parts will do it, and today I will teach till codominance, and after that we'll stop. Your dihybrid cross in the next class. Okay, dihybrid cross in the next class. It's fine, students. It's fine. We need to understand. See, I can rush all this. I can finish this entire chapter in 2 hours. Entire chapter in 2 hours. But if I do that, God promise you will not understand. You right now, all of you have an advantage. You have time right now. So students, if you think all the time in the world, if you take right now and focus on understanding concepts, it will benefit you in long run. Okay, so understand, students, understand. Okay, understand and learn right now. No need to buy heart any of them. You have time right now. Let's not do one shots right now. We'll do one shot later on. Okay, students, here is the question. Students, can I get the answer very quickly? Students, can I get the answer very quickly? I don't have time now. Okay, I'll teach you, Amrita. I'm coming there. Don't worry. Students, I'm coming there. Next session will be next week. Because students, tomorrow I'm doing one shot living world. All of you know that, right? Tomorrow I'm doing one shot living world. Tomorrow morning I'm doing one shot living world. So please watch that also. Morning 11 o'clock tomorrow. Okay, students, what is the answer? What is the answer, students? What is the answer? We can't waste time in questions. Okay, students, very easy, students. 2 or alternate forms of a gene is called as an allele. F1 cross with the homozygous parents is called as your test cross. Cross of F1 with your any of the parents is called as back cross technically. Number of chromosomes in set is called as your ploidy. Oh, this is a very good question. Students. In a plant, black seed color capital B is dominant over white seed color. So this is dominant phenotype, recessive phenotype. In order to find out the genotype of black seed, so in order to find out the genotype of your dominant phenotype, to find the genotype of your dominant phenotype, okay, with which of the following genotypes will you cross it? Basically, they're asking you, what do you do in test cross? Basically, they're asking you, what do you do in test cross? What do you do in test cross? That's what they're asking you. What do we do in test cross, students? What do we do in test cross? All of you. In test cross, do we take dominant parents or recessive parents? We take double recessive parents. Yes or no? We take recessive parents. Yes or no, students? All of you in the chat. In test cross, what do you, students? In test cross, we take this capital B. Capital B, this is your dominant phenotype. We cross it with recessive parents. Recessive parents are going to be what? Capital small b, small b. That's all. Yes or no, students? Recessive parents. Small b, small b. Okay, students. Now, shall we start with income dominance? Okay. Now, students, I want all of you to understand what exactly happens in income dominance. First of all, very simple it is. Okay. In income dominance, what is happening is, let's say we have two different alleles. One is dominant, one is recessive. But in income dominance, what is happening is, this dominant allele is not dominating only. This dominant allele is not working very well at all. And this recessive allele is there, no. Recessive allele is expressing itself. So basically, try to imagine a classroom. In that classroom, backbenchers are not backbenching. Backbenchers are very quiet. But so when backbenchers are very quiet, frontbenchers can also talk in that class. So it is a class where the front benchers are also talkative. Students, the front benches are also very, very talkative, students. That is called as your test score, students. In test, sorry, that is called your incomplete dominance. In the case of your incomplete dominance, one allele is incompletely dominant over the other allele. That's all. Students, try to imagine mother is a doctor. Mother is a doctor. Father is an engineer. Mother is a doctor. Father is an engineer. But the son is a musician or a dancer. Okay. Son is a student. So here what happens is that the F1 generation is there, right? F1 generation will not resemble both the parents. Will not resemble both the parents. It's like father is an engineer, mother is a doctor. I will be a dancer, student. I will be a dancer here. The F1 generation will not resemble both the parents. It is very unique creature we have, students. Here example is a dog flower, also called as atrium species. Example is it happens in your dog flower, also called as your students snapdragon. Students, very fancy name, no? Snapdragon, also called as your atrium species. One more example is your 4 o'clock plant also. 4 o'clock plant also. That is Mirabilis jalapa. Okay. Mirabilis jalapa also it happens, students. 4 o'clock plant also it happens here. Okay. It happens, students. Okay. Now, students, let's just do a cross right now. Let's do a cross. Okay. Now what is the first step? First step is the parents. First step is the parents, students. Now, what do we have in the parents here, students? In parents, we have the one is red flower crossing with white flower. Okay. Here, red flower is capital R, white flower is small r. Okay. Now, next step is gamete formation. Students. In gamete formation, what do we get? Capital R here and small r here. Gametes. What are the next step, students? Next step is selfing of F1. Next step is F1. Sorry, students. Sorry. Next step is F1. First filial generation. What do we get? We take this, we get capital R small, students. Technically, this was supposed to be red because of capital R here. Capital R was supposed to be dominant, but this capital R is not dominant, it's not working really properly. This end up becoming pink color. It is not red, it is not white, it is somewhere in the middle. It becomes pink color. Okay, students, it is called as pink color. It becomes pink color, students. Okay, it becomes pink color right now, F1. Now, next step is called as what? Selfing of F1. Selfing of F1. Okay. Here, what do? We take capital R small r, cross it with capital R small r. Okay. Let's do the cross, students. Let's do a cross. Okay. Capital R, small r. These are the gametes we get here. This is going to be capital R, capital R, capital R, small r, capital R, small r, and small r, small r. Here, this is going to be completely red color. These two are going to be incomplete dominance, just like your F1. They are going to be pink color. These two are pink color. And unfortunately, this one is going to be white color. Now, students, if I ask you what is the phenotypic ratio here? Phenotypic ratio is basically how a plant looks from the outside here. So, one is red. Students, one is red, two are pink, and one is white. 1:2:1. Okay. Now, if I ask you what is the genotypic ratio here? Genotypic ratio, students, is going to be what? Homozygous dominant, heterozygous incomplete dominant, and homozygous recessive. So, students, can I tell here, in this case, phenotypic ratio is equal to genotypic ratio, just like your test cross, 1:1 category also. So, yes or no, students? Yes or no? Yes or no? All of you. Yes or no, students? Here we have phenotypic ratio and genotypic ratio, both are equal to each other. And students, here is a image of your snapdragon. You see, pink color flower is there. There is somewhere white is there. Yes or no? These are pink color flowers here. There we also get pink color flowers here, students. Pink color flowers. Okay. We also get pink color flowers right now, students. Pink color flowers right now, students. All of you, please, if you're able to understand, smash the like button, students. Smash the like button right now. Okay. Now, students, next step, here is a simple question. Here is a question, students. Solve this question. Very simple question, right? A pink flower snapdragon plant was obtained. So we got a pink color flower, we got, okay? A pink flower snapdragon plant was crossed. Okay? Pink color flower is capital R, small r. Okay? It was crossed with red flower. Crossed with red flower. Okay? Snapdragon. What type of phenotypes are expected? So from this, what is the phenotypes do we expect, students? Let's do a cross. Capital R, small r, capital R, capital R. Okay, it's supposed to be only one capital R only because only one is there. So what do we get? So what do we get here? We get one is red. Not one. Half of them are red. Half of them are pink. So what is the answer? What is the answer? Red flower as well as pink color flower. Yes or no, students? Yes or no? Yes or no, students? Red flower as well as the pink color flower. So what is the answer, students? What is the answer? All of you? What is the answer? Answer is 1. Students, do you find that questions easier or not? This is a NEET 2024 question. NEET 2024 question right here. Incomplete dominance, students. Incomplete dominance. So 50% are red, 50% are pink. Okay. Students, today I'll finish in next 15 minutes. I'll finish. Don't worry. Next 10 minutes I'll finish. Start solving, students. Start solving. I'll be drinking some water. Tanni. I'll be drinking some tanni. No, tanni. Students, we call tanni only in Tamil. Students, what do you call this in Telugu? Water in Telugu? In Tamil, it's tanni, I feel. Tanni could we say. Also, next. Done. Sorry. See the question, students. I just want to move aside. Sorry. Read the question. Very simple, easy. Very easy question, students. Nero. Nero is in Kannada, I know, sir. You're telling me Kannada only Nero. I know Nero. Oh, Telugu also Nero, students. Okay, Nilu. Okay, Nilu. Machi Nilu. Manchi Nilu. Okay. Students, how many of you are coming to Hyderabad this weekend? How many of you are in Hyderabad this weekend? We are coming to Hyderabad. We are coming to Chennai also. Chennai, last time I visited, it was beautiful. I had enjoyed in Chennai big time. I came to Annanagar itself, Chennai. Right. Hyderabad, first time ever, students. First time ever, I'll be coming to Hyderabad. First time ever in my entire life, I'll be coming to Hyderabad. First time. So this weekend, right? Okay, students. Students, obviously the first option, right? Law of segregation does not apply here. Students, we know law of segregation is what? It is universal. Law of segregation is universal. All of you should know. Law of segregation is universal. Yes or no? It is universal. Now, students, this I will teach you next class because it will take some time here. Okay, I'll teach you, students. Very easy it is. Right? Right. Students, it's very, very easy. Law of concept of dominance, one small paragraph is there. It only tells students, law of concept of dominance basically tells you that there are two alleles, dominant allele, recessive allele. Recessive allele is because of certain modification. Recessive allele is because of certain mutation. That's all. Okay, they're given a simple example here. Okay, now let's say we'll take one allele is giving some type of enzyme. One allele is giving some type of enzyme. Okay, some type of enzyme, students. Okay. Okay, some type of enzyme here. So this enzyme can be normal or less efficient, non-functional enzyme, no enzyme at all. So it is normal, it can be dominant. These two are basically recessive. Students, the whole point of concept of dominance is that this dominance and recessive concept came into existence because there was some allele which was modified. Some allele was mutated. That is why we have a recessive allele. That is why we have a recessive allele, students. We have a recessive allele. Okay. I'll teach you codominance. Wait, no, I'll teach you codominance. Okay. Now, students, shall we start codominance, students? Shall we start codominance? Students, if you look at this image only, can you tell me what is codominance? If you look at this image right now itself, can you tell me what is codominance, all of you? This image, student, this image right here. Can you tell me what is codominance? Yes or no? Students, I'm thinking even dihybrid cross also, but I will stop at codominance itself because I've gone very slow today. I've gone very, very slow for all of you. Okay, students, what exactly happens in the case of codominance, students? In the case of codominance, both alleles are expressing. They're like, I will also not keep quiet. You also not keep quiet, students. Try to imagine a class. Try to imagine a class which is filled with only backbenchers. Students, try to imagine a class where front is also backbencher, back also is backbencher. So I will become mental there. Okay, so in codominance, both alleles are expressing here. Now here, students, the F1, right, students, let's imagine, right, students, imagine father is an engineer, mother is a doctor, the son is a doctor and engineer both. The son is doctor and engineer both right now, students. So the F1 here, the F1 generation will resemble both the parents. It will resemble both the parents, students, both the parents here. Now here one concept, students, here one concept arises here. The concept is called as multiple allelism. Okay, students, till now I have told you one gene has two alleles, but that is not true actually. One gene can actually have more than two alleles. Yes, one gene can have more than two alleles. That is concept is called as multiple allelism. Students, the concept is called as multiple allelism. So in the case of students, in the case of your multiple allelism, one gene can have more than two alleles. That is called as multiple allelism. Now here, in the case of human blood grouping, right, human blood grouping, there is something called as I gene. This I gene actually has three alleles. It has IA, it has IB, and it has small I here. Now even the students, even though there are three alleles here, do you think these three alleles will be present in your body? All three are present in my body? No. Even though there are three alleles for this I gene, only two will be present in your body. So out of three alleles here, two are present in us. Students. Okay, present in us, students. Now here, this is a typical example. This is a typical example of your ABO blood grouping. ABO blood grouping, students. So ABO blood grouping is an example of codominance. ABO blood grouping is an example of codominance. ABO blood grouping is an example of multiple allelism also. Both is true. It is also example of multiple allelism. So if you students, yes or no? ABO blood grouping is an example of your multiple allelism. ABO blood grouping is an example of your codominance. Both are possible. Now students, do you want to know something extra here? What makes this IA, IB and small I so different from each other? What makes them different? Students, understand. All of you know, students, this is your RBC, RCB or RBC? RBC, not RCB. Okay, RCB on the mind. Okay. Now, this RBC right now, it has plasma membrane on top of it. Yes. On top of this plasma membrane, right, there is a sugar present. And this sugar is different in your A allele. This sugar is different in your IB. And in the case of small I, there is no sugar at all. No sugar in small I. Okay. Students, in the case of your IA, here, A, here can you see, students, the outer sugar, this one is the outer sugar here. This outer sugar is called as your N-acetyl galactosamine. N-acetyl galactosamine, students. But in the case of IB, sure, what do we have, students? Here we have basically galactose here, galactose. In the case of small I, no sugar. No sugar at all, no sugar. So because of this sugar, it makes them very unique, students. Because of this sugar, it makes them very unique. Now students, tell me what is your blood group in the chat. We'll do your blood grouping now, students. All of you, what is your blood grouping? Who will volunteer, students? Who will volunteer right now? Who wants to volunteer? Okay. All of you, who wants to volunteer? Okay. Let's take, students, tell me your real name. No, what is AIMS? I know college is there. I know you want to go to college, but tell me your real name. In my class, you are not just a random student to me. In my class, you are a valuable student who is attending my class for the very first time, and this student should be happy after my class. So tell me your real names. All of your real names. Yes? Okay. Okay. Students, let's take Darshini. O blood grouping. Okay, we'll take O later on, students. I need someone else. I need someone. KG Music, I need your real name. I will not take your real name, students. I need real name. All of your O, students, I'm also O. But this O gang is here. Huh? The O gang only is here. O gang. What is this? Huh? Only O gang is here. Okay, students, let's say random person who has B blood grouping. Okay. B blood grouping will have IB or IB small I. In this case, dominance, B blood grouping. In the case of A blood grouping, we will have IA or IA small I. Small I will not express. In the case of AB, this is the best part. This is where the codominance happens. IA is dominant. IB is also dominant. So both are dominant here. So both will express and we get AB blood group, students. We get AB blood grouping here. Okay, we get AB blood grouping. Both are expressing here, students. Both are expressing. Now, students, all the O gang in the chat, students, all the O gang, all of you. In case of O positive, we do not have any dominant allele, students. In the case of O positive, we do not have any dominant allele. We only have recessive allele, students. We have recessive. Students, don't worry. They are universal donors. We are universal donors. Okay, we can donate blood to all of. I will give blood to you, you give blood to me. Okay, so we are universal donors here. Okay, student, that is your human blood grouping here. Human blood grouping. You can see here also the same thing is given here also, students. Same thing is given here also. Okay, students, the inheritance of coat color in rabbit is also codominance. Please, students, please remember that. It is also example of multiple allelism actually. Multiple allelism. Okay, multiple allelism. Students, here is the question, quickly answer. I hope it is easy for all of you now. Where's my O gang? O gang, answer this. Lalita, nice. O doesn't have any sugar, students. O doesn't have any sugar. Students, all of you quickly smash the like button. There are only 92 likes. Make it 100 as soon as possible. All of you, make it 100 as soon as possible. All of you. Make it 100 as soon as possible. All of you. Students, what is the answer? What is the answer, students? What is the answer? Students, how was the class? Students, how was the class, Bhagya? How was the class? Students, I'll tell you why. I'll tell you why. You should comment in my videos. When there's a new student who's attending a class, a new student will never look at the lecture. First, a new student will always look at the comments. So it's your job to share the knowledge now. So when you comment on this video, the other students will realize this video is useful to everyone. So all of you need to comment on this video. This is your job. Your job is to comment on the video. So once you comment, everyone will get to know that, oh, this is a nice lecture. Then they will also watch my lecture. They'll also be happy. So sharing is caring. Okay, that's what answer. What is the answer, students? Students, one is the answer, students. One is the answer, yes or no? Students, it says when A, IA and IB are present together, right? They express same type of sugar? No. One A is producing what? IA is producing what? N-acetyl galactosamine. IB is producing galactose. So they are not producing the same sugar. They are not producing the same sugar at all. Okay. They are not producing the same sugar at all, students. Same sugar, students. So students, in the next class, I'll be starting with starch synthesis in pea plant. Okay. So next class, I'll be starting from here. In the next class, I'll be teaching with this dihybrid cross, as well as, students, as well as the entire chromosomal theory of inheritance. Until sex determination, I will teach you in the next class. Until sex determination, next class. If possible, I will finish the entire chapter in next class. If not possible, we'll go 3 parts. Okay. Okay, students. So students, with that, we come to the dramatic end of today's class. I hope all of you students, I hope all of you enjoyed. Students, thank you so much. Now students, before I go again, yes, all of you, I will show you cash. I'll show you my dog. My dog is waiting always, students. Okay. Students, I want all of you to quickly go to the description here. Students, smash the like button first of all. Make it 100 as soon as possible, students. Make it 100 as soon as possible. You can go here. You can click here. Okay. You can fill your information here and we will be coming to your city. So please let us know if you're from Hyderabad or from Chennai. Please let us know that. Okay. So when you fill this form, we get to know that you're coming to meet us. If not, we'll cancel. If not, we'll cancel only. I'll not come. I'll not come then. Okay. So please let us know by filling this form where you're from. Okay, students. So thank you so much, students. Bye bye. Take care, all of you. I will see you in the next class. Until then, keep studying. Let me know in the comment section, students. The question is, students, did Mendel, what is a pure line? Is pure line homozygous or heterozygous? That is a question to all of you, students. Pure line is homozygous or heterozygous? That is your comment question to all of you. Okay. Pure line is homozygous or heterozygous, students. Okay. Pure line is homozygous or heterozygous in the comment section. I will check all the comments. I will reply to all the comments. So in the comments, students, in the comments, all of you. Okay. Okay. Oh, 10th class, oh, fancy. Okay. So let me know in the comment section.